Hello everyone. Welcome to DrOmics Labs Private Limited. Hope you are all doing well. Myself, Chitra Raskar, I'm a bioinformatician. So, first of all, thank you for being here today. I'm delighted to have the opportunity to discuss one of the most revolutionary, fascinating, and powerful techniques in molecular biology, and that is RNA sequencing. So, over the next few minutes, I will share with you the details of RNA sequencing basic concepts, workflow, and pipeline. So, here we are going to discuss how we can analyze the data. We will explore how RNA sequencing has transformed our understanding of gene expression. But before we dive into these details, let's take a moment to appreciate the importance of gene expression in biological processes. So, what is gene? We know that it holds the instruction for life, and how they are expressed and regulated plays a crucial role in development, disease, and everything in between. So, traditionally, studying this gene expression constitutes significant challenges. However, with the advent of RNA sequencing, we now have a powerful tool that allows us to analyze the transcriptome. So, what is transcriptome? It is nothing but the complete set of RNA molecules in the cell or tissue. So, RNA sequencing has empowered scientists to unravel complex biological processes, identify novel genes and isoforms. And here we can, by using this technique, we can gain more knowledge. We can gain insight into disease mechanisms. So, without further delay, let's start this journey. So today here we are going to discuss the content, that is nothing but first I will explain you about the NGS, what is next generation sequencing, different platforms and its applications. Next, we will talk about the RNA sequencing introduction and some important concepts in the NGS. Then we are going to discuss about the workflow of RNA sequencing, means which tools we are going to use here for the analysis, and then how to do this downstream analysis study. That in details we will see today. So let's move further. So now let's understand why this technology has invented. So as we know that cell is the basic unit of life. So life it is coming from, or we can say originated from life only. So let's take one example. If there is a male and female, so there is a sperm and egg present, and until and unless this sperm and egg won't fuse, there won't be another life. So scientists also wants to know what is the reason behind it. What is the thing which is creating a life? So it is nothing but the DNA, and that DNA it is present on the chromosome. So on that DNA, there are four nucleotide bases present. Those are the ATGC. So if you want to know that how this nucleotide base is present on DNA, we have to do the sequencing. Okay. So for the sequencing, there are different instruments or we can say platforms are available. So let's see here. You can see we are having first generation, second generation and third generation. So let's talk about NGS here. So this NGS technique it is used to determine the order of nucleotides in an entire genome or the targeted regions of the DNA and RNA. And here we can detect abnormalities across the entire genome. This technique it is faster and it gives us a accurate sequencing result. But now we know that there is a previously Sanger sequencing has been used widely, right? So what is the difference between this Sanger sequencing and next generation sequencing platforms? So basically, Sanger sequencing can sequence only single DNA fragment at a time. But this is not the case with the NGS. Here, NGS it is massively parallel. It sequences millions of fragments simultaneously per run. So this process translates into sequencing hundreds to thousands of genes at a time, millions of genes at a time. So that is the main difference between Sanger and NGS. And in the third generation, we can use advanced instruments such as PacBio and Oxford Nanopore. But you know what? These tools, all of these platforms works differently and they are having different read size. So that I have mentioned here. So if we check the read sizes here, Illumina provide approximately 100 base pair read size. But if we compare here, you can see for the nanopore sequencing, it is having higher read size, around 500 base pair to 2.3 MB. Unless it is provided by the solid, that is approximately 50 base pair. Actually, Illumina it is widely used nowadays, but you can see previously this Roche 454 and solid was in use, but now currently commercially they are not available for the sequencing. So now let's see exactly how we can do this NGS, how we can perform the NGS here. So let's take the example of this human DNA. Okay, like we have isolated the DNA here or RNA here. So once we isolate the DNA, we have to convert this DNA into small fragment. So now why we have to convert this DNA here? Can we sequence the whole DNA directly? So the answer is no, because there is no sequencing platform is available which can read the whole DNA. So that's why we have to do the fragmentation by using some enzymes. So once we do the fragmentation, act- we have to send the sample for sequencing. And this is one of the biggest disadvantages in NGS nowadays, that there is no sequencing platform is available. So here for this fragment, we will ligate the adapters, we will prepare the library and send the sample for sequencing. So after the sequencing, we will get the result in the form of reads. Actually, these reads are nothing but the fragments only, but technically we call them as an reads here, where we get to know what is the sequence. Then we have to merge all of these reads together to make the whole genome back again. So for this purpose, we are going to use different assembler tools here. So now at this stage, we get to know that at which position what base it is present there. So next step is we have to predict the genes from that genome. So for the gene prediction, there are different tools available such as Proca, Prodigal, Augustus, Eugene, GeneID. Okay. So once we get this gene information, we can submit this information to different databases such as NCBI, SwissProt, KEGG, Motif, PDB, USS. You can check out others also. So now I wanted to talk about the different applications of NGS. So we are having two types of samples here, DNA sample, RNA sample. So when we have the DNA sample, we can go with this applications, and when we have the RNA sample, we can go with this one. So under DNA, we are having DNA sequencing. So why we do this DNA sequencing? If you want to study variations, such as SNP type of variation, insertion, deletion, that type of variations, you can check it in the DNA sequencing. In the whole genome de novo assembly, we can study genomic features. We can find out the gene information. In the chip sequencing, we can study DNA protein interaction. Here you can find out histone information also. And in metagenomics, we can study the diversity amongst different organisms, or we can say from the community of organisms. And in methyl sequencing, it is very famous for the gene silencing study. And if you are interested to study the methylated region, you can go with this methyl sequencing. And in the mRNA sequencing, here also you can study the gene silencing. But you can see now methyl sequencing coming under the DNA section. And here for the mRNA sequencing, we have to use the RNA sample. And here we can, in the methyl sequencing, we can study disease association also. And now let's talk about reference base and renome base RNA sequencing. So here we can study the gene expression. So in details, we will talk about the reference base RNA sequencing. So what is RNA sequencing? It is nothing but the technique to determine presence and quantity of RNA in a sample. Okay, and this technique, it is also known as differential gene expression profiling, or we can say digital gene expression profiling. So why it is important to study this RNA sequencing? So basically because it tells us that which gene it is or turn off in the cell, which gene it is highly upregulated or downregulated in the cell. And due to this, it helps us to understand the basic biology of the cell. And if it is indicating any disease, that information also we can retrieve here. So I just mentioned that it is also known as an differential gene expression. So why it is called it as differential gene expression? Because we are checking the expression of genes in different types of samples. So whenever it comes to the differential, at least we require two samples here. Maximum number of samples you can take any. So what are those samples? Those are nothing but your control sample and treated sample. Control is your normal, healthy, not disease sample, and treated is abnormal, healthy, or we can say disease, or if we have provided any kind of treatment to that sample. So now next is steps in the NGS. So in that we are having three steps: library preparation, sequencing, and data analysis. So in details how I will explain you this later in the bootcamp. So now next is types of RNA sequencing. So here we are having two types of RNA sequencing: reference base and de novo base. So in this reference genome for that organism, it is already available in different databases and with their annotation files also. And in the de novo base, in the name itself, it's a de novo. So if you are having any novel organism, for that novel organism, you won't be getting any information in any databases. Okay. So in details, we will see about this reference-based RNA sequencing. So this I will explain you with the help of one figure. So just give me one moment. Yeah. So now let's consider here we are having four samples which contains the reads, that is control 1, control 2, treated 1, treated 2. And let's consider this data, it is from the Homo sapien organism, and all of this raw data we are getting from the sequencing company. Okay. So now here as I mentioned, let's consider this data. It is from the Homo sapien organism. For that Homo sapien reference genome, it is available in the databases, different databases such as NCBI, UniProt. You can take it from anywhere, and you can see here with the reference genome, we will get the annotation files also, and those annotation files are present in GTF and GFF format. Okay, so let's consider now we downloaded the reference genome for the Homo sapien. But now to study the differential gene expression, we have to map or align all of these reads to this reference genome by using some tools. Those tools I will explain you. I will tell you about these tools in details during the pipeline explanation. Okay. So now you can see on this reference genome we are having different genes, or we can say transcript, as we considering this as an RNA. Okay. So once we map this reads on this reference genome, let's consider 5 reads got mapped from this control 1 sample. 10 reads got mapped from this control 2. And there is no read it is mapping from this treated 1 sample. And from the treated 2, 20 reads are mapping here. So what I mean by 5 reads are mapping from control sample, it means that 5 times this G1 gene it is expressing in control 1 sample. Okay. And 10 times it is expressing in control 2, and 20 times it is expressing in treated 2 sample. So in a conclusion, I can say that this G1 gene it is highly expressed in the treated 2 sample, as more number of reads are coming from this sample. Okay, and if this treated 2 sample it is from any disease, we can say that due to the higher expression of G1 gene, that disease it is occurring here. Okay, so this is the overview of reference based RNA sequencing. So now further, we will discuss about some important concepts here. So first concept is analysis plan. So before performing any experiment, we have to decide this biological analysis plan here. And here, what is the exact meaning of this biological analysis plan? Here we can check how we wanted to see the expression of genes. Okay, means here we just saw that we are having two types of sample, control and treated. So whether you want to check the expression between control treated, like control versus treated and treated versus control. But in details, what is the difference between this control and treated sample? We will discuss in detail in boot camp. Okay, which is going to held on 3rd and 4th June. So next is types of sequencing. We are having two types of sequencing, single end sequencing and paired end sequencing. So let's talk about single end first. So here at the time of sequencing, sequencer can read the data only from one end at a time. So that's why this technique, this sequencing, it is called it as a single end sequencing. And it can be either from forward or reverse side. And as an output, we can get only one file here, forward or reverse. Next is paired end sequencing. So whenever sequencer is reading the data from both side at end, okay, means sequencer is reading the reads from both side, forward and reverse. So as an output, we can get two files here, forward and reverse. So next concept is depth and coverage. So depth is nothing but how many number of times a specific site, or we can say particular nucleotide, it is readable by sequencer. So that is what the depth, and this depth we have to decide. We have to tell the sequencing company that we want this much depth, that how much we have decided. So for the RNA sequencing, minimum depth should be 50x and maximum you can put any number there. But just keep it in mind, when depth increases, cost of the sequencing also increases. And next is coverage. So coverage means how many number of fragments are able to cover your reference genome. So if we are having more number of reads, it is covering more of our reference, and that can gives us a good result. So when will our coverage will become high? When we have high number of reads, and when we have the high number of reads, our depth will be high there. So we can say that this depth and coverage, it is directly proportional to each other. Okay. So next concept is Fred quality score. So this Fred quality score, it is assigned to each and every base which is predicted by the machine. Means here we can measure the quality of nucleotide bases generated by the sequencer. Means how much accurately our base has predicted by the machine. That information we can get it here. But here also, as it's a quality score, it is having some cutoff, and that cutoff, it should be greater than or equal to 30. Means I will explain you here why we decide to set the cutoff as 30. If we are having Fred quality score 10, it means that we are getting 90% accuracy. If our Fred quality score is 20, we are getting accuracy as 99%. But if it is 30, we are getting 99.9%, which is good. So that's why we have decided to set it as 30. Means 1000 times our base has predicted by the machine, but out of 1000, only one time it has predicted incorrectly, and 999 times it has predicted correctly. So that is good only. Okay, so that is the meaning of Fred quality score. Next is mapping quality. So here in Fred quality score, we just discussed that it gives us the quality of bases. Now in mapping quality, we are deciding how our read it is aligning to the reference genome. So next is BAM or SAM file format. So BAM and SAM file format, these are the mapping file format. When we get the mapping result, that result we get it in either BAM format or SAM format. And it also depend on tool, which tool we are using. Some tools can give us the result in BAM format, some tools can give us the result in SAM format. So BAM stand for the binary alignment map format. And as it is the binary, So it is in computer readable format, and SAM it is in the sequence alignment map format, which is the human readable format. But about this SAM format in details we will discuss in the bootcamp. Next is FASTQ format. So FASTQ format, it is a read format and it contains 4 lines. So that I will explain you here. You can see we are getting these 4 lines here. So in the first line, it always start with the @ symbol and it contains the sequence ID. So this sequence ID is our read ID. Second line contains the actual sequence of the read. Third line contains some special symbol, but most of the time it is the plus symbol. And the use of this third line, it is nothing but just to separate the second line and the fourth line. And in the fourth line, we are getting some characters, numbers, some symbols. And this here we call this as an ASCII characters. Okay, so this ASCII characters is it represent the Fred quality score. Means it has this Fred quality score, it has encoded into ASCII characters. But in details, how this encoding has happened, how this conversion uh, is done by the tool, that we will discuss in detail in bootcamp. Okay, so now let's talk about the pipeline. So this is just the overview I have mentioned here. First, we are having control sample and treated sample, and then we have to isolate the RNA, we have to prepare the cDNA, and then we have to do the fragmentation. And for that fragmented cDNA, we can ligate the adapters here. And then we will send the sample for sequencing, and after the sequencing we are getting the FASTQ files. In this FASTQ files our reads are present. Okay, now how we can analyze the data by using the bioinformatics tool, that we will discuss here. So first step is that quality check. We have to check the quality of our reads. Okay, so for that quality check we are going to use the FastQC tool here. So now I'll show you how the result looks like and what are the important parameters we have to consider, how we can differentiate whether our read quality it is good or bad. So let's see. Yeah, so you can see now here this is our FastQC report and we are having some parameters here: basic statistic, per base sequence quality, per tile sequence quality. But we are not going to consider all of these parameters. There are only 3 important parameters we are going to consider. Per base sequence quality, it is first. So you can see now here in this per base sequence quality, it shows us the quality according to each and every base. So if you see now here on this x-axis, it shows us the position. Okay, and on this y-axis, it shows us the Fred quality score. And we are getting some yellow boxes here. So these are nothing but the box whisker plot. So this also in detail we will discuss later. But now you can see when this box whisker plot, it represent bases. So when our bases are falling under this green area, we are having a good quality of bases. If it is present on this orange area, we are having a moderate quality of bases. And if these bases are falling under red area, we are getting a bad quality of bases. Okay, then we are having overrepresented sequences, but currently we are not able to see any sequence here, so I'll show you another document. So in this overrepresented sequences means these sequences are overly present. It is present multiple times in our data, and if this sequence it is present in our data, it can lead us to misinterpretation of the result. But you can see now, I will tell you one thing. If in this possible source we are getting no hit, we don't need to remove this sequence. But other than this no hit, if you are getting any word like adapter, primer, then surely we have to remove those sequence. So you can see now here Illumina paired and PCR primer given. So surely if this type of sequence present in your data, we have to remove this. And now adapter content. So if any adapter sequence it is present in your data, we have to remove that, and you will get the graph for that if it is present with the different colors. But now I just told you that we have to remove those sequences, but how can we remove it? We have to use different tools here. Okay, that is our next step. If any overrepresented sequence or adapter sequence is present in our data, we have to remove those by using cut adapt or trimomatic tool. So our next step is trimming and cleaning of data. Okay, so after this, once we remove these sequences here, again we have to check the quality of our data. So again we have to perform the fast QC, and then we are good to go for the mapping. So mapping of reads on genome. So for this mapping, we are going to use tools such as BWA, Bowtie2, HiSet2. You can use any one of these. And then this mapping result, we can get it in the BAM or SAM format. But if we are getting result in BAM format, we won't be able to understand. Only computer will understand. So what we have to do? We have to do the conversion of BAM files into SAM format. Okay. And with the help of that SAM format, we will be able to get the count of reads here. Okay. But how we can get the count of reads? For that purpose, we have to perform the cufflinks here. Okay, and once we get the count of reads, means how many reads are mapping from control sample, how many reads are mapping from treated sample on the reference, that exact count we will get it from the cufflinks tool. But to quantify the differential gene expression, to calculate it, we have to merge all of those count together from both samples. Okay, so merging of reads, that we can do by using the cuff merge tool. And the next step is calculating differential gene expression. For that purpose, we can use this curve tool here. Okay. So now you can see here, we are having, after this, we have to study the downstream analysis. So let's see. So after analyzing this whole RNA sequencing data, there are several downstream studies we can perform to understand the transcriptome, or we can say gene expression in a sample. We can go with differential gene expression analysis, functional annotation and pathway analysis. So in this differential expression analysis, this involves comparing the expression levels of genes between different samples, such as disease and healthy sample, treated and untreated sample that we just discussed, right? And in functional annotation and pathway analysis, this involves mapping differentially expressed genes to know the biological pathway and functions which are related to the biological processes. And for this purpose, we can use databases such as Zoodeprot, Panther, ShinyGeo, K also we can use. Then alternative splicing analysis. So this RNA sequencing data, it can be used to study alternative splicing. Means it is nothing but the mechanism that generate multiple protein coding transcript from a single gene. Okay. Then we can study identification of non-coding RNAs such as microRNAs, non-coding, long non-coding RNAs, and we can study this non-coding RNA role in gene regulation and cellular processes. In transcriptomic assembly, here it is used to assemble the transcriptome of an organism, or we can say cell type. We can provide a comprehensive view of that expressed genes and their different variants. Then we can study epigenetic analysis also. So here RNA sequencing data can be integrated with epigenetic data. Means we can study DNA methylation, we can study histone modification, and we can try to understand the interplay between gene expression and epigenetic regulation. Then next is single cell RNA sequencing. So this RNA sequencing data, it can also be obtained at a single cell level. Okay, but it allowing us to understand the gene expression on the individual level of cell. Means we can study the identification of cell subpopulation. Okay, so now these many downstream studies we can do here, and it can provide insight into gene expression patterns, biological processes, and many more like cellular functions, and it can inform the development of new therapeutic strategies, or we can discover the potential drug targets also. And now you can see here, I have mentioned one image has represented here. So we can generate the heat plot, and then we can generate the whole kind of plot also, heat map also you can generate. And for this purpose, we can use the programming languages such as R programming language. With the help of Python also we can do this. So for these services also we are providing here. So if you are interested, you can check on our website. So now, as we wrap up this presentation, I hope that you have gained a deeper appreciation for the power and potential of RNA sequencing. So it is a tool that continues to evolve with advancement in technology and analysis methods, and it is constantly pushing that boundaries of what we can uncover here. So in the end, if you want more information about accurate and reliable RNA sequencing services, please visit our website. We are more than happy to be assistance. Thank you so much.